This is chapter 18 on gene expression. Just because genes exist in a cell's genome, all the genetic material of a cell, does not mean they are always expressed. What does that mean? That does not mean they are always transcribed and translated into a product. Gene expression is when certain genes are turned off or turned on depending on the type of cell, as well as the necessity for the product of that gene to actually be made. So you do express some of your genes in some of your cells and not others in other cells. So one example here is the SRY gene on the male Y chromosome is only activated once during early embryo development when it's needed to create male genitalia in an unborn fetus. Another example is your skin cells. Even though they contain your entire genome, they're definitely going to be producing melanin. It's a pigment to protect your skin. However, your nerve cells would not produce melanin. They contain the genes for it, but they're not expressing that gene in those cells because it's not necessary. Bacteria often respond to environmental changes by regulating whether or not they do transcription. Two examples are the trip operon and the lac operon. Those are the two we're going to learn about. And these are in bacteria. E. coli, which lives in our gut, synthesizes the amino acid tryptophan. You may have heard this one before because it's a common amino acid in turkey. It's actually the one that they suggest makes you sleepy. And it synthesizes that from precursor molecules, so other things that build it. There are actually 5 genes that code for subunits of the enzymes needed to synthesize tryptophan. So what that means is that there are 5 genes that make the enzymes that make tryptophan. That's what that's essentially saying. It doesn't actually make tryptophan itself, it makes the enzymes that make tryptophan. These 5 genes are clustered together in the E. coli genome. The genome of E. coli is a circle, and as a single promoter, and a single promoter serves all 5. Some typos here. 5 genes together. These are called a transcription unit. Recall from our last chapter that a promoter is the site where RNA polymerase binds to DNA to begin the transcription process. Remember, transcription is going from DNA to mRNA in the nucleus. So this is just a little schematic of this. These 5 genes all make enzymes that ultimately will synthesize or make tryptophan. And tryptophan is something that is needed to build your proteins. Interestingly, if you're making tryptophan, negative feedback, which we're going to learn about later in the year, will cause these enzymes to not be made because you're already making tryptophan. You won't have to. There'll be a negative feedback here. So we mentioned the trip and a lac operon. What is an operon? Transcription of these 5 genes gives rise to one long mRNA, and that's going to code for 5 polypeptides. So those are strands of amino acids that make up the enzymes needed for E. coli to synthesize tryptophan. Each gene still has a start and a stop codon, so that each polypeptide is still separate, but they're transcribed together. Grouping the genes together means that one transcription unit can be operated by one on or off switch. The switch is what we call the operator. Together, the operator, the promoter, and the genes are what we consider an operon. You can think of this kind of like the lights in your house. One light switch may turn on multiple lights. And that's the idea here, that one operator is going to turn on and transcribe multiple genes. The trip operon and the lac operon are 2 that we're going to discuss in the E. coli genome, which affects us because E. coli lives in our gut. Okay, so the trip operon is what we consider a repressible operon. That's going to become important later. By itself, the trip operon is turned on. Okay, what does that mean? That means that at any given time, RNA polymerase can bind to the promoter and transcribe the gene. So it's typically being transcribed. However, it can be turned off or stop transcribing it by something called the trip repressor. The trip repressor will bind to the operator and block the attachment of RNA polymerase to the promoter and prevent transcription. So the repressor prevents transcription of the genes. And thus, if we don't have the transcription of the enzymes necessary to make tryptophan, we are not going to make tryptophan. Here is an idea of what this kind of looks like. So this is the repressor. Okay, the repressor is actually being made separately. This is the trip operon here. It consists of the promoter, the operator, and the genes for the enzymes that will synthesize tryptophan. So here is your mRNA piece that ultimately will be translated into those enzymes. RNA polymerase binds the promoter. The operator is typically not being blocked by anything. So on a normal basis, this is being transcribed. We are making tryptophan. So tryptophan's not present. You don't already have it. The repressor is inactive. Notice this is an inactive form. And the operon is on, meaning we are making tryptophan. Well, we're actually making the enzymes that make tryptophan, but you get the point. The trip repressor is a protein product of a regulatory gene called TRPR, which is located some distance away from the trip operon and has its own promoter. That's what we were showing over here, is that this repressor protein is actually made separately by this particular gene. And that's just the same picture again. Why is the trip operon not switched off permanently? The binding of the repressor to the operator is reversible. Repressor is an allosteric protein which has two shapes, active and inactive. And the trip repressor is synthesized in an inactive form, which with little affinity for the trip operator, meaning it's not going to attach to the trip operator very easily. If you go back and look at it, its shape is not really fitting. Only if tryptophan binds to the trip repressor, the protein changes to an active form that can attach to the operator, turning the operon off. Let's see how that works here in just a second. Tryptophan functions as a co-repressor because it cooperates, that's where we get co from, with the repressor protein to switch the operon off. So as tryptophan accumulates, so for example, say you eat a lot of turkey and you got lots of tryptophan in your gut, that bacteria is then going to have tryptophan. More tryptophan molecules will associate with the trip repressor and they will bind to the trip operator and it will shut down the production of the enzymes that make tryptophan. If the cell's tryptophan levels drop, transcription of the genes will resume. So several hours after your meal, tryptophan goes down and you start resynthesizing it. So notice, remember, this was the repressor. Tryptophan will actually bind to the repressor. That will turn it into an active form. Now see, this is going to fit in that operator. If that operator is full, or it's got the repressor in it, then RNA polymerase cannot bind to it, so we are not making the enzymes that make tryptophan. The trip operon is then what we call repressible operon because transcription is usually on. Normally, it's on, but can be inhibited with a molecule, tryptophan, when it binds to that repressor. This is again just a kind of a blown up picture of it. You accumulate tryptophan, It binds to the repressor, turns it into an active form. The active form shape matches really well with the operator, and RNA polymerase cannot bind, thus no transcription. We're not making the enzymes that synthesize tryptophan. The lac operon is kind of the opposite idea. This is actually an inducible operon. An inducible operon is usually off, but can be stimulated to make that particular product. For example, when you drink milk, the disaccharide lactose, remember it's a milk sugar, becomes available to the bacteria E. coli in your colon. Lactose is digested into the monosaccharides glucose and galactose by the enzyme beta-galactosidase. Only a few molecules of this enzyme are present in the E. coli cell growing in the absence of lactose. However, if lactose is added to the environment, say you're drinking a big glass of milk, then the number of beta-galactosidase molecules will increase drastically. The gene for making beta-galactosidase is part of the lac operon, along with two other enzymes that function in breaking down lactose. So here's kind of the opposite situation. You have an active repressor that fits perfectly. RNA polymerase cannot bind. You're not making any product. In this case, that product is beta-galactosidase. So no lactose, repressor's in its spot, operon is off. However, if you have something called allolactose, which is actually an inducer, that will bind to the repressor. It'll cause it to go to an inactive form. And in that case, it's not going to bind. And RNA polymerase will bind. And it will go ahead and synthesize these enzymes, in particular beta-galactosidase, which will then allow lactose to be broken down. So you drank a big glass of milk, lactose is present, repressor's inactive, not working, operon is on. So it's just the opposite is the trip operon. The regulatory gene for this operon is called LAC-I, which codes for the allosteric repressor protein that can switch off the lac operon by binding to the operator. And let's see, that's right here. Remember before where we showed that there was something that made the repressor? Well, this is the same idea. This gene is responsible for making this active repressor. Or I'm sorry. Woo, my bad. It is responsible for making it's responsible for making the repressor. The lac repressor is active by itself, binding to the operator. And switching the lac operon off, an inducer inactivates the repressor. So it's just the opposite as a trip. The inducer is allolactose, which is very similar to lactose. In the absence of lactose and allolactose, the lac repressor is in its inactive form, and the genes for lac operon are not expressed. Sorry, this should say active form, because it would be in its active form here. No lactose, repressor is active, fits into the operator, RNA polymerase cannot bind, no enzyme being made. So this is considered an inducible operon. When lactose is added to the surroundings, allolactose will bind to the lac repressor, alter its shape, hinder the repressor's ability to attach to the operator, and thus transcribe the gene and produce the product, in this case, the beta-galactosidase. Drink milk, the inducer inactivates the repressor, can't bind here, RNA polymerase can bind, transcription happens. So the difference between repressible versus inducible operons, the repressible enzymes generally function in anabolic pathways, which means when you're synthesizing something, you're making something from smaller pieces. The inducible enzymes generally function in catabolic pathways, in which you're breaking down something into something else, something into something simpler. By producing enzymes only when needed, it helps the cell to save energy. There is no reason to be producing tryptophan if you have tryptophan in your food. If you're already eating it, there's no reason. There's also no reason to make an enzyme that breaks down lactose if there's no lactose present. That doesn't make any sense. So really, it's all about avoiding wasting energy. So this is a little bit different. This kind of talks about a different side of things, not about whether or not you're making the whether or not you're, well, I'm always trying to rephrase this. This is not about whether, this is, instead of being about whether you're making or transcribing it, it's more about how fast you're transcribing it. Okay? So let's go through the example of the lac operon, but this is not just about being able to transcribe the beta-galactosidase. This is about how quickly we're doing that. E. coli prefers to use glucose as an energy source and only uses lactose when glucose is in short supply and lactose is present. So how does E. coli actually sense that? How does it know that glucose is not there and lactose is? Something called cAMP. It's called cyclic AMP, adenosine monophosphate, something like that. Accumulates when glucose is scarce. So we don't have a lot of glucose, so cAMP is going to accumulate. cAMP binds to something called CAP, which is an activator. And CAP assumes its active shape in order to attach to an area upstream of the lac promoter. This attachment increases the affinity for RNA polymerase for the promoter, and it stimulates gene expression. So here's our cAMP. It's going to bind to our CAP. It's going to turn that active. It's going to bind upstream of the promoter, and then RNA polymerase is going to bind and transcribe. So this is when lactose is present, but glucose is a little bit scarce. So our cAMP, or cyclic AMP levels, are really high, and then we're going to make lots and lots of the lac mRNA because we have lactose present, so we need to synthesize a lot of something that would break lactose down. How does E. coli sense this? If the amount of glucose increases, the cyclic AMP concentration will fall. So more glucose means less cAMP. And the CAP will detach from the operon. RNA polymerase will bind less efficiently. So it's still binding, but not as efficiently to the promoter. And it will, transcription will happen, but it will happen at a lower level than before. So when we have this cAMP, it's actually going to increase the affinity for RNA polymerase to bind. If we don't have the cAMP, then we have less affinity or efficiency. It will still make the enzyme, but not as quickly. So here's the inactive CAP. So we don't have no or very little cyclic AMP. So RNA polymerase is going to bind, but a little less likely. The state of the lac repressor determines whether or not transcription takes place, and the state of the CAP controls the rate of transcription. So the state of the lac repressor, whether or not it takes place, the state of the CAP is going to control the rate, rate of transcription. It's kind of like the operon has an on and off switch, but it also has a volume control. Okay, it's going to determine the rate of that transcription, not whether or not it happens, the rate of which it happens. And then we're going to stop for this one, and then we'll get to eukaryotic gene expression in part 2.